Step 1. Sample concentration using CeleMag Clean-up Bead. This video contains Celemics Enhanced Hybridization Kit protocol with bead concentrated sample. For single plex, please refer to Step 1, Concentrate of Sample using CeleMag Clean-up Bead in your manual for detailed protocols. For multiplex, please refer to Step 1, Pre-capture Multiplexing of Samples in your manual for detailed protocols. Step 2. Enhanced Hybridization of the library. Preparation of blocking reagent with DNA library. Transfer 10 microliters of pre-capture multiplex sample to a new 8-strip PCR tube. Add 5.6 microliters of blocking reagent to each sample tube. Mix well, seal, and spin down the tube. Keep the tube containing the blocking reagent with DNA library on ice before hybridization. The total volume of the tube is 15.6 microliters. Preparation of hybridization buffer and probe mix. Make sure that the hybridization buffer and high buffer enhancer are pre-warmed at 37 degrees before use. If any precipitates remain, keep incubating at 37 degrees until the solution is clear. Prepare the hybridization buffer and probe mix described below. Mix well with gentle pipetting. Transfer 25 microliters of hybridization buffer and probe mix into a new 8-strip PCR tube. When using the 96 well plate, do not add the hybridization buffer and probe mix on the 96 well plate at this step. Keep hybridization buffer and probe mix tube at room temperature before hybridization. Set the program into a thermocycler. Place the blocking reagent with DNA library tube or plate on the thermocycler and start the hybridization thermocycler program. Keep the tube on the thermocycler until procedure 3F. Once the temperature reaches 65 degrees, which is the thermocycler step 2, additionally, place the hybridization buffer and probe mix tube into the thermocycler and incubate at 65 degrees for 1 minute. After performing procedure 3C, which is incubation at 65 degrees for 1 minute, open the thermocycler and remove 2-strip PCR tube caps or sealing film. Transfer 15.6 microliters, the entire amount of blocking reagent with DNA library, into hybridization buffer and probe mix tube or well of 96 PCR plate. Mix well by pipetting up and down 2 to 3 times. Seal the tube tightly with a new cap completely and start thermocycler step 3 of the thermocycler program. Step 3. Preparation of CeleMag Streptavidin bead. Preheat wash buffer 3 at 70 degrees in a heat block for the next step. Step 4. Selection of the target captured library. Vortex CeleMag Streptavidin bead to achieve a homogeneous state. Add 50 microliters of CeleMag Streptavidin bead to a new 1.5 ml low-bind tube. Wash the CeleMag Streptavidin bead as follows. Add 200 microliters of wash buffer 1 to the tube containing 50 microliters of CeleMag Streptavidin bead. Mix well on a vortex mixer and spin down briefly. Place the tube in the magnetic separator until the solution is clear. Keep the tube in the magnetic separator and discard the supernatant. Repeat steps A through D twice to a total of 3 times. Add 200 microliters of wash buffer 1 to the bead and proceed immediately to the next step. Step 4. Selection of the target captured library. Keep the sample hybridization mixture from Step 2, Enhanced Hybridization of the library, at 65 degrees and immediately transfer the sample hybridization mixture to the bead solution. Invert the tube 3 to 5 times to mix them thoroughly. Rotate the sample on a rotator at 25 RPM for 30 minutes at room temperature. Spin down briefly. Place the tube on a magnetic separator until the solution is clear. Keep the tube on the magnetic separator and discard the supernatant. Take the tube out of the magnetic separator and add 500 microliters of wash buffer 2 to each tube, 180 microliters for a PCR tube or 96 well plate. Mix well on a vortex mixer and pulse spin down. Incubate the sample for 15 minutes at room temperature. Briefly mix the sample on a vortex mixer and pulse spin down. Place the tube on the magnetic separator until the solution is clear and discard the supernatant. Rinse the bead with preheated wash buffer 3. Add 500 microliters of preheated wash buffer 3 to each tube, 180 microliters for PCR tube or 96 well plate. Mix well on a vortex mixer and spin down briefly. Incubate the tube for 10 minutes at 70 degrees in a heat block. Briefly mix the sample on a vortex mixer and pulse spin down. Place the tube on the magnetic separator until the solution is clear and carefully discard the supernatant. Repeat steps A through E twice to a total of 3 times. Spin down the tube for 3 to 5 seconds and place the tube on the magnetic separator to discard residual wash buffer 3 completely. Add 30 microliters of nucleus-free water to each tube and mix well on a vortex mixer. Spin down briefly. Now, on-bead capture pool is prepared. Step 1. Amplification of the captured library. Prepare PCR master mix described below. Add 50 microliters of the homogeneous on-bead capture pool to the PCR tube. Mix well and spin down briefly. Place the tube that contains 50 microliters on a thermocycler and run the PCR program. Step 2. Purification of sample using CeleMag Clean-up Bead. Vortex CeleMag Clean-up Bead to achieve a homogeneous state. Add 90 microliters of CeleMag Clean-up Bead to a new 1.5 ml low bind tube. 0.2 ml PCR tube or 96 well plate is also available. And the amplified capture DNA library of 50 microliters to the tube. Mix well using a vortex mixer and pulse spin down the tube. Incubate at room temperature for 5 minutes. Place the tube on the magnetic separator until the solution is clear, which is about 3 to 5 minutes. Keep the tube on the magnetic separator and discard the supernatant. While keeping the tube on the magnetic separator, add 500 microliters of fresh 80% ethanol to each tube. 180 microliters for PCR tube or 96 well plate. Incubate for 30 seconds at room temperature, then discard ethanol. Repeat wash again, step 7 and 8. Spin down, place the tube on the magnetic separator, and discard residual ethanol. Air dry the sample tube at room temperature for 2 minutes or until residual ethanol evaporates completely. Add 32 microliters of nuclease-free water to the sample tube and vortex to mix. Pulse spin down and incubate for 2 minutes at room temperature. Place the tube on the magnetic separator for 2 minutes until the solution is clear. Transfer the supernatant of 30 microliters to a new tube.